Well, welcome everyone. I'd like to start by thanking the organizers for allowing us to do this symposia. So as the title suggests, we're going to talk about space, the final frontier in pharmacology and physiology. I was speaking at a conference about a year ago on mitochondria, and one of the things that I sort of took away from that was this notion of what the limitations are. A lot of inventiveness, money, resources are going into building hardware, but there's very little still input into figuring out how to get humans to sit inside that hardware to get them up into space. And I think this is going to lead to some really amazing moonshot moments where new things are going to be discovered, new pharmacology, new ideas. And so in light of this, what NASA had decided to do is create a 10-site NASA twin study. So 10 sites across the US were chosen to take on various aspects of human physiology and adaptation to a year-long event in space. And the caveat to this was that it was a monozygotic twin pair. One twin stayed on ground, the other twin went up in space, and lots of data was collected on them before, during, and after space flight. You'll be hearing from 3 of these investigators today on what was discovered and what the potential of this is. So without further ado, we'll get started with the first speaker for the session. It's Chris Mason from Cornell.
All right, well, good afternoon, everyone, and thank you very much for the introduction. It's a pleasure to be here and be selected by the organizers and to put together this panel. So, as you've seen and heard, we're going to walk you through some work in my lab as well as collaborative work with some of the other scientists, esteemed scientists on the panel, as well as collaborators around the country and the world that we've been taking some lessons from the twin study, and I'll summarize them to you, but then how we've been now applying them to other missions that are happening with SpaceX and Axiom and other spaceflight providers. So, jump right in. I also have some conflicts of interest to disclose, which is work I've either co-founded or been compensated in some way for, listed here below. So, more than ever, it's worth noting we are a spacefaring species. If you look at what happened since 1957, when Sputnik first went up into space, you can see there was mostly the US and Russia that were launching objects and crews into space. And then things began to change in the early 2000s where more launches were occurring, getting up to several hundred happening per year. But then in the past couple of years, things have really transformed. And so not only are there more things being put into space than ever before, but more countries are doing it. And now it's actually a super exponential increase in the number of objects and crews going up into space. And what this means is we have more opportunities to fly objects into space. In fact, last year was the record holder for the most number of launches of things put into space. So it really changed the trajectory of spaceflight. And actually this year so far is on track to beat last year. So really an extraordinary time for humanity and also some of the science, and eventually I'll tell you some of the aerospace medicine that's happening in flight. There's even architectural firms who you can now plan out what your moon base might look like, or a lunar laboratory. This is a rendering from a firm that's actually building out some of the plans for a European Space Agency for their lunar habitats, which you can't, quite live at them yet or get sort of lunar beer, but you can start to imagine this and actually become a lunar architect is a legit job. There's also plants that have been grown on the moon. This is from the Chang'e 4 mission, where you can actually have cotton growing on the moon. Again, you can't quite get your lunar cotton or your lunar genes quite yet, but at least we know it's possible to start to grow actually Earth-based plants, in the sort of lunar gravity environment. And this is, an exciting time not only because of what's being put on the moon, but also 4 different space stations are being planned and built as we speak. So by 2029, there might actually be 4 distinct space stations orbiting around our planet, which again is an extraordinary feat of technology and really perseverance and some of the market forces to create these opportunities. And there's also a bit of a new space race. So it used to be Russia and the US, but now is a little bit of a space race between the US and China. China wants to put boots on the red planet by 2033. And the US might be by 2037. It keeps putting push back, but the space race has really been heating up between China and the US to get to the moon and then to Mars. And so what this means is we have this extraordinary world we live in where we have many opportunities, a lot more missions, a lot more data, and we want to get to the moon, get to Mars. You can see here, this is a rendering from NASA. It almost looks as if Mars here is sucking all of the atmosphere away from the Earth and taking it over. But that's just a rendering. It means we need science and technology to get to Mars and ideally become a multi-planetary species. But the biggest question that we have in our lab, as well as what question that you heard before that NASA really has, is if we're going to be on the moon and Mars, how do we monitor people's health and how do we keep them safe? How do we ensure they can survive the trip and make it back home? So these are all really big questions in terms of how do we make sure that we can keep the crews safe and also have them not just survive, but thrive on these other planets. So as you heard before, this was the first chance to look at this question in great depth with NASA, was with the NASA twin study, where we had Scott Kelly here on the right. Mark Kelly says he's the handsome one with the mustache, but they are identical twins, and they had a chance to actually follow them over the course of a year when Scott was in space for a full year. And Mark Kelly is now Senator Kelly, so we have the beginning of maybe the senatorial genome project in our laboratory. We could look at genetic differences between Republicans and Democrats, and maybe you could CRISPR a Republican into a Democrat or modify them potentially. We haven't tried that study. That would not get an IRB approval, but we do have the beginning of some of the senatorial changes in flight. And so when Scott Kelly went up into space for a year, this is a picture of the rocket when he was heading up into space. He did a lot of things in space, including taking a lot of photos, taking blood samples, stool samples, urine, metrics, cognitive experiments. He took a lot of beautiful pictures like this and posted them on Twitter and Instagram. He also would do some juggling of fruit there in space, but that's actually not that hard to do in space because it kind of stays in front of you. But these, great work he did on biomedicine as well as for some entertainment. He grew a zinnia flower when he was up in space. It was kind of great to see this as well. And then, after about a year in space, he undocked from the space station, and we were all anxious that day because he was coming back to Earth. He was bringing a lot of the samples back with him. He was, it was the end of a year-long mission. We were curious, how would he respond after coming back after such a long time in space? The longest time at that time for any American astronaut. But the chute deployed just fine. He was making his way back to the atmosphere and then landed in Kazakhstan at that time, where we had a lot more work in Kazakhstan, and was helped up by some of the other crews and NASA staff and gave us a thumbs up and seemed to be doing fine. But then this is a video recently released of the first time that he was actually trying to walk after he'd been in space for a year. So you can see he's walking okay, he's mostly doing all right, but then of course it's not as easy because he hasn't used his body, even with all the strength training, really in 1G for about a year. And so it was hard on his body. So he had some slight difficulty first walking, but he also had really unusual inflammation that occurred. So actually his skin felt like it was on fire, he said. He had rashes and discoloration anywhere that he had contact, including just the weight of clothing alone was enough to create a rash on his arms and his legs. And he really felt like he wanted to go to the emergency room. He had flu-like symptoms. It was a very challenging time in his body. And the big question we had as a 10-study team was just why? What happened in his body? How do we learn from the stress on his body of what happened over a year and build out profiles that could help us guide future missions and maybe look at countermeasures and maybe potentially drugs that could target them. And so in that end, we wanted to look at a really complex and in-depth study of every molecule we could, get down to a genetic and cellular and molecular view of the change after a year in space. And so you can see what we looked at was almost everything that we could. So this included genetic variation, epigenetic changes, RNA expression, proteomics, chromatin, antibody titers, B cell, T cells, telomere length, cytokines, metabolomics, microbiome, cognition, and vasculature over the course of about every 5 to 6 weeks, over about 2.5 years, to measure what we could in terms of what had happened inside of his body. So some of this has been published, so I'll go through some of the quick highlights really quickly. And so in particular, one of the sort of the flagship paper we published in 2019 was to look for clusters of biology that changed in concert. Because when you see things move up and down at the same time across multiple layers of biology, that often indicates co-regulation or some coincidence of how the body's responding to a stress. And so particularly worked for things, for example, that increased in flight but then came back down. And so some things, of course, we had many things that changed. Metabolites changed, microbial species, cytokines would spike up, some proteins changed, but telomeres also got longer. And you'll hear a little bit about this more in the next talk about how and why telomeres get longer, because we've done a lot of follow-up work on that with Dr. Bailey. But we could see that was one surprising feature, but most things did come back to normal when he got back to Earth. And so we could see, for example, even though he got sort of younger because his telomeres got longer, which are the caps at the ends of chromosomes that can shrink often with age, and he did gain 2 inches, but they both went back to normal when he got back to Earth. So it is not the fountain of youth to go in space, or if it is, you have to stay in space the whole time. And so what other things that we looked at, and you'll hear a little bit of this more in Dr. Bailey's talk, is we even saw some of these same telomere elongation and cytokine spiking for people that climb Mount Everest. This is with Matt Moniz, who was climbing this mountain with Willie Benegas, and we had a twin stay at sea level, and one twin actually go to the top of Mount Everest, and also confirm that with this study as well, which was a fun exercise to see. But to get back to the rash question, one of the things in terms of, for this audience, is we looked at the really pro and anti-inflammatory markers that changed the most, and looked basically in this paper from Gertz et al., what are all the drugs that could target each one of these specific interleukins or C-reactive protein, interleukin receptor antagonists, for example, or CCL2. And you can see here, for most of them, they're very flat compared to Scott, who's in blue, and Mark, who's in green. And they stayed flat or they're somewhat elevated in flight, but it was the day that he landed that we got sometimes hundreds of percent or thousands of percent increase above baseline. And so to answer the first question of what happened to his body in terms of the stress, these are the markers that were the most stressed. And what's interesting is IL-6, for example, when we first started seeing COVID patients at our hospital, we saw the same thing. So actually I have some emails from postdocs in my lab saying, I feel like we're looking at the twin study again because the astronauts and COVID patients exhibited some of the same systemic stress for IL-6 in particular. And so this is interesting to see how these connect. And we published a list of all the drugs that could target them. But the challenge is you don't necessarily know if these are responsive to just spaceflight. Do you want to turn off these pro or anti-inflammatory markers, or is this part of the body's adaptation? And so this is something that was one of the bigger questions at the time. And we said, when we published this and published several follow-up papers, we said it was overall good news that all these layers of biology mostly return back to normal. But it's clear we needed more data. We had an N of 1, N of 2 if you include the control, so it was very limited. And we tried to compensate for that by taking as many samples across as many layers as we could, but we still need more data. And we want to put into the context of how often do we see these same signatures. So we launched an IRB and actually a full informed consent process at Cornell. Actually, if any of you are going to space and would like to enroll in this study, please come see me after the talk. We have open enrollment for the study. We don't have that many people, but we do have some. And so it's only $50 million these days to go to space. It's not too bad. But we launched, building on the twin study, a Space Omics and Medical Atlas IRB, which, as you can see here, has a broad range of biophysical, molecular, and cellular assays that we run off controls, as well as any astronaut that go up before flight, in flight whenever possible, and also post-flight towards at least 2 years, in most cases 5 years of a post-flight study. So this includes everything from blood work to plasma to stool, urine, saliva, buccal samples, and many, many layers of omics data, which I'll walk you through shortly, because in all instances we're trying to learn from these crews and compare this to what we saw for the twin study. And you can see here, my student who made this figure couldn't resist making stool brown and urine yellow, but I think that's fine because that's about what they look like. So in any case, the first chance we got to use this IRB was with the Inspiration 4 crew, who worked closely with the SpaceX medical operations team to plan out this study, and really had the chance to use 4 all-civilian crew that were heading up into space in 2021. So this was actually really fun because it's the first time I got to go to see a launch, which if you've never been to see a launch, I highly recommend it. It's actually, you can kind of see this calm atmosphere near Cape Canaveral. They load the rocket up, and actually as it's starting to launch up, it happens sort of in 3 phases, where you first will see the rocket light, because of course light moves faster than sound, but then you will feel it actually in the ground because sound moves faster through the earth than it does through the air. And then you can actually hear it as it sounds like the atmosphere is ripping as the sort of rocket goes up into space and makes its way up there. It's a very beautiful launch. I was very happily able to go see one of them. And so when they got up to space, they were actually going up to, on average, 575 kilometers, which is higher than the Hubble Space Telescope, higher than the International Space Station. So experiencing a little bit more radiation than you'd see in the ISS, and in general, a pretty high elevation space flight. And I'll come back to that later because some of the upcoming missions are going to go even farther out away from Earth. And when they got up in space, they took some selfies because they're in space, and you get excited, you'll take some selfies. But they did also then get to work, collected over 700 different swab samples, collected blood samples, did a portable ultrasound, did some other work here. Still had a smile on their face though, but did a lot of collections and work, and then made their way back to Earth. So they splashed down. Actually, it was a short mission, only a 3-day orbital flight, and then splashed back down here. And you can see how they're all cheering and very excited because they didn't die, basically, when they landed. They're very excited to have made it back alive and very happy. And then the Dragon capsule made its way up into the boat, and we had some teams of our researchers helping to get the HEPA filters and swabs and get started right away on the work. So what we did is, I'm going to walk you through, so this battery of multi-omic studies, and then whenever possible, I'll show you results comparing it to the twin study. But one of the first things we did was what's called a multi-ohm profiling. So you can actually take individual single cells and look at open and closed chromatin. So you can see how is gene expression being regulated, and is chromatin being compacted and often turning off genes, or is it open and more accessible? And we can also measure expression on a cell-by-cell basis. This is the protocol from a company called 10x Genomics. And once you actually prep the library, you have single nucleotides that you can isolate, you barcode, and actually have a transposon that hops into DNA if it's open and creates the library. If it's closed, then it won't access it and won't create the library. And the expression is from a reverse transcriptase. And so once we do all the preps, then we sequence and compare to the human genome. And what we found was I did 150,000 different cells for gene expression and epigenetic changes. And on the left, you can see if you take each point as a cell, we can see these clouds. That's a UMAP plot, which is basically like a Rorschach plot for genomics. You can see these differences between cell types, though, where you can see natural killer cells in blue, B cells you can see at the bottom in red. We can see T cells, other variations of CD4 and CD8 T cells in the middle. And on the right are some of the QC metrics, but mostly these are what we'd expect from PBMCs or peripheral blood mononuclear cells, fairly just a control step, and what we expected to find were these kinds of cells. But what's really interesting is this slide is that we then were able to examine what genes are changing expression, so differentially expressed genes on the top, and genes that go up are in orange, and those that go down are in purple. We can see this wide symphony of different changes of how the cells respond to stress, which is not surprising. If you eat spicy food, if you go for a run, if you come to a conference, your genes will be changing their expression in a circadian or stress-related fashion all the time. So that wasn't too surprising, but we could see some core sets of features where a vertical line is a gene and each of the horizontal lines is a cell type, so T cells, B cells, et cetera. And we can see some consistent signatures of change for all across all cells, as well as some that are distinct to each individual cell. But the most striking feature to me from this data was, or in these data you can see at the bottom, the monocyte cell population, that big bar of green, means that it's opening dramatically its chromatin, whereas most other cells, the gray means nothing had changed. It was very stable. So you could actually see, in this case, this wide adaptation to spaceflight from monocyte cells, more so than any other cell type. This is really striking because we figured most cells have a distinct response to any stress, but in this case, the monocyte cell populations had the most dramatic response by far epigenetically compared to any other cell type. And then we want to see, well, how long does that last, right? If you change, just if you compare pre-flight and post-flight, how long did it last on the day that they landed, so immediately post-flight at these differentially expressed genes versus differentially accessible regions, or DARs, and how did they persist? Did they go away after 6 months? And we can see most things go down. The body does continue to recover and return to baseline, but the day they land, of course, is a bit more stressful, as we saw with the twin study. But the cells, again, the CD14 and CD16 monocyte cells are the ones that are by far the most perturbed, and actually even post-flight, 6 months, still have some changes relative to where they were pre-flight. And so we're getting more blood drawn actually in 2 weeks for the crew again to continue to monitor to see how they'll change over time, to put this in context of just other stresses on the body. So there we go. So the other thing we wanted to look at is, okay, here we go, is we want to compare, this is kind of a large, okay, compare this of what immune cell types are coming up, and this is just a dense plot to show you for each of the different cell types in these columns, where do we see upregulation in red or downregulation by different pathways. And we can see things like UV response or response to DNA damage, or basically UV response, again on the top for different cells, and inflammatory response, things that we expect to find, and we've seen again and again across the twin study, and we see again here for these same pathways. And the other thing we want to look at those, with the twin study, we just of course had 2 males, but we had males and females in this mission. And we wanted to see male versus female, who has more changes in the cell types. Again, it's just a handful of subjects, but it's interesting to look at. Females so far have far less changes that persist actually baseline or post-flight, whereas males have, across all cell types almost, have a broader change that takes them longer to return to baseline. So this means for future missions, we're recommending that there should probably be a parity in the crew, or at least or more females if you have to pick, because they do seem to recover faster, at least in terms of these metrics. Now, the Artemis crew that's going to the moon does only have 1 female, so we're pushing NASA to include more females if they can in future missions, because they seem to recover a little bit faster. So the other thing that we looked at across all this gene expression data was other pathways that might be related, again, to the twin study. Particularly in epigenetics, we could see a lot of differences here. This is the enrichment for biological process on the left and the reactome on the right. And we did also see telomere stress and DNA damage again pop up on the right. And you'll hear a little bit more from Susan Bailey in the next talk about what we looked at there, which included some telomere length. And so that's a preview of what's coming next. The other few things we looked at, of course, in the crew was not just the human cells, but they, of course, have microbial cells. And we wanted to look at what happened to them when they're a tiny dragon capsule, what happened to their body, right? So we were inspired by a study, one of my favorite studies of a microbiome study. Jessica Green did a study where before and after roller derby, they swabbed the skin of the players just to see if you have a really, kind of rough and tumble rugby game, or, in this case, if you're, doing any kind of roller derby, your microbiome becomes more similar to your opponents after the study. So we wanted to see the same thing essentially here for these crews. Do we actually see the microbiome of the skin get more similar in this tight crew? If you go into an elevator today during the conference, you might, if it's a very tight elevator, you're probably exchanging microbes with your other elevator partners. Maybe you don't want to think about that, but I think that's fascinating. You should just be aware of it. But for the crew, we did these 10 locations to see what would happen. And actually, indeed, we could see on the right first that the difference between them went down, so became more similar on the right, with very large error bars, but the average in general went down in the flight and then after the flight. And then we also looked at what happened for just across different areas of their body. Where do we see the most species? Where's the most microbiome diversity? And you can see on the left that the armpit actually won the award for the greatest diversity. So you might look at your armpit and think it's a cauldron of despair and darkness and dangerous organisms, but it's actually this wonderfully rich rainforest, a tapestry of diversity and new species. And so next time you're in the shower, you should be excited by your armpit, I think, in terms of its possibilities. Or at least it was for this crew in this case. So we also want to compare the crew. We did metagenomics and metatranscriptomics, looking at all the DNA and all the RNA of what's changed across these 10 regions before, during, and after spaceflight. It's from a paper that's in review from a postdoc, Braden Tierney. And what we could see is we saw many, many changes, and I showed you some of those, but the other thing we looked at is we looked to see which species changed the most with all those, basically the immune cell changes I just showed you, and see how many, which of the cell types here by different colors, how many associated microbes do we have. In some cases, we had a handful for bacterial microbes versus viral, for the nasal and oral different sort of microbes we found. But the skin, actually other parts of the skin up to the body, had many, many more correlations between the microbes that were shifting their abundance and matching what we could see for some of the genes that were changing in the immune cells. So trying to look at the exchange of the immune system and the microbes on their skin, you could see there's the list of the associated microbes. And we can't say they're causal, we can say they're associated. So let's look at that a bit more closely. We then also did a skin biopsy of the crews, because we wanted to look at what's changing in the vascular, what's happening on the skin and the microbiome. Again, to get back to this question of the rash and disruption to the body, what's happening. So what we did is we looked at molecular and cellular changes, some of them I just showed you. We wanted to understand epithelial barrier changes related to the microbiome, and also look at the immune changes, looking at peripheral blood versus what we could see basically from spatial data, transcriptome data, as well as proteomics data, which I'll show you shortly. So what we did is use the geomic spatial transcriptome method. If you're not familiar with this, it's where you can actually have essentially barcoded antibodies that will bind to specific, essentially these probes, and where you can actually have a UV light that cleaves them off, and then you can sequence them, or you can use even qPCR. But this is the different crew members. We did skin biopsies before and after spaceflight, and then probed with 18,676 gene probes to see what was present for spatially what's happening in their skin. So not just gene expression going up and down, or even by cell type, but this spatial profiling lets us see in situ where are we seeing changes in gene expression. And we look at outer epidermis, inner epidermis, the dermis, and vasculature for each one of the crew members before, during, and after spaceflight, actually before and after. And you can see some of the labeling we had, for example, for the DNA DAPI staining and pan-CK. You can see the structure of the skin here, and then we quantified across those 18,000 genes. So then we compared to those 150,000 cells I was just showing you before, and compared basically what do we see for what's happening in the skin, these regions of interest on the geomics. You can see we see these overlap of these differentially expressed genes, where we could start to see what's happening in the skin mirrors what's happening for the genes actually in the blood. Actually, in some cases, particularly on the day of landing, that R+1 is the return of one day after they landed. We could see a lot of the overlap showing up, particularly on that day. But also, we saw these other genes, these are the 12 genes that we saw consistent response. But for the most part, they were very different responses, which is not too surprising. The skin is a different environment than the internal blood system, of course, but we do see some signatures that match to both. And then we also want to see, based on the sequencing of blood, what else could we do to sequence it and characterize it, and not just get individual small fragments of RNA, but do what's called direct RNA sequencing. And this is another way to profile the blood. We actually can take, let me go back to this, you can actually take individual molecules of RNA as they move through an nanopore and sequence them natively. So we don't have to make cDNA or convert them. You can sequence the RNA as is. And we take blood RNA and fragment it and sequence it. You can see we can get thousands of nucleotide molecules that can read the full length of the transcript. rather than just getting a small bit at the end of the transcripts or a few hundred bases. So we can actually see on the right, we get these full transcript lengths. And what we can see is we also not only see the bases, but we can also see modified bases. Using a tool called M6ANet, we can look at methyl-6-adenine, or the modified bases of RNA, kind of like epigenetics says methylcytosine of the DNA. This is the M6A, which modifies the function of RNAs. And we can see the expected DRAC motif when we did motif search. And from this data, we can confirm a lot of the same expression and methylation genes that we're changing, mostly in hematological functions. So you can see some of the genes for actually deal with how the body's creating new blood cells, also some of the HLA alleles we can see pop up, and sort of on the right side, you can see the different pathways and the methylation change. It's a very dense slide, but I'll just show you at the top is mostly the hemoglobin and sort of heme regulatory genes that are changing both their expression and their methylation pattern, which is what we saw from the single cell data as well. So the last vignette I want to share from you from this study, and then to compare to the twin study is, what do we see for the I4 crew and look at plasma and also can do proteomics and metabolomics, but then also look at extracellular vesicles and proteins and peptides, or EVPs, and see what's different between these sort of micelles that are in the blood, these exosomes, or those some people just prefer to say EVPs, but basically small encapsulated vesicles that are in the bloodstream, and did proteomics and metabolomics of them as well. And so what we could see is that, again, to compare to what's in plasma versus, say, in exosomes, or what's detected in both, you can see at the top of the Venn diagram, there was a fair amount of overlap, but we did also see some distinct changes. So we could see, for example, ERK1, ERK2, MAP kinase cascading that would show up more in plasma, but less so in EVPs, which is more looking at hematological regulation, like blood coagulation, hemostasis, and sort of other heme-related functions on the right here. So there seem to be some different molecular profiles if you look within the bulk plasma versus exosomes, and so we could see some of these differences here. And the thing that's most, I think, striking about this study is we did see brain-related proteins enriched in the secretome, as things in the vesicles, after spaceflight. And we had seen a little bit of this actually in Scott Kelly when he got back to Earth, so there were some brain-enriched peptides in his exosomes. But this represents the first sort of confirmation study, where if you look at what pathways are enriched, if you look by proteins, we can see it's sort of, a lot of it's dealing with synaptic plasticity, dendrite morphogenesis, and synapsis, and actually the brain has an enrichment you can see on the right in terms of immediately post-flight, more so than long-term space flight. So we actually see the day they land, it seems to be that there's this just potential disruption of the blood-brain barrier, or some changes of brain-derived proteins showing up in the blood. And what those are, you can see here, some of them, for example, are SNCA, or we can see neurogenin, or CNP, for example, that in red, they spike up the day that he lands, but they mostly return to normal. But we can see these are interesting because it indicates that there's not necessarily, damage, but maybe disruption to the blood-brain barrier, because these should normally be found as brain-associated proteins and maybe as enriched in the cargo that's coming from the brain, which could have implications for longer missions or heading to Mars. And so, okay, so that's what we've seen for some of the twins data. That's what we've seen actually from these brain-derived proteins and other molecular profiles. But what's next? So the other thing we're doing now, of course, is merging a lot of these data with the sort of other human spaceflight data. So, for example, on the bottom, you can see the overlap with the NASA twin study bulk RNA-seq, and we see a lot of the same red and blue patterns show up. If you compare to the blood samples we did from the twin study, we're seeing a lot of the same genes, showing their change in expression in CD4 and CD8 and NK cells, much like we did in the twin study. So we're seeing significant overlap there. And we're seeing also some of the overlap between twin study data and the other transcriptomics data up on the top, and comparing all these data to each other. And then we're also looking a little bit for the different, basically countermeasures and ways we could target these genes, these consistently dysregulated genes or cytokines. We've been looking at sort of links as well as connectivity map and the drug bank to try and see which genes would target some of these specific molecular changes. Now, it doesn't mean you want to try this drug necessarily, but it means you could at least get a list of potential countermeasures to look at. So this is actually work that's also in review, is to see which ones actually could have a target. And we have 148 drugs that have been found from the compound database at Drug Bank that match these gene expression profiles. So we think we could begin to at least start to think about countermeasures and do some of this work in situ and in vitro first, if we can. The other thing is we're launching later this year is an aerospace medicine biobank, so that you can request samples as well as the data, akin to like what you see for the UK biobank, is that this will be any spaceflight associated samples. You can actually request samples and data and access it to begin to help some of your own research. And again, if any of you are going to space, please let me know after this talk. And then finally, we've been working with NASA's Gene Lab Repository, which is an omics archive where all the data can, whenever it's been fully consented, is open for anyone to download, process, and analyze and compare to some of their own data. So if you're looking at the effect of spaceflight in response to a different drug or a different trauma, all these data, you can actually submit queries, Jupyter notebooks, and also request samples and engage in collaborations with NASA's Gene Lab Repository. And so what's also coming up next is we did just get samples in the last few weeks from the Axiom 2 mission. This was actually for about a 14-day long mission in the space station. So this was the crew here. This actually featured the first Saudi female astronaut to ever go up in space, and also, you know, the second and third ever astronauts. So really exciting to see a broader diversity, more parity in the crew, so we can look again at more male-female differences, which we'll be looking at shortly. And also, we're planning now for the Polaris Dawn mission, which is coming up later this year, or maybe should be later this year. The final date's to be announced soon. But this is the Polaris missions. We're going to have 3 different missions with some of the same crew members from the Inspiration 4, which Jared Isaacman is going to then go on 3 more missions. So we have the same person that's gone up into space 4 times total by the end of this mission. And the last one will be the first human crewed flight of the Starship, which is, of course, here what will take us, in theory, back to the moon and potentially to Mars, and can carry at first 12 people at a time, but eventually 100 people at a time can fit into the Starship for some of these larger missions, which really will fundamentally change the game as how you can do spaceflight research and in general get crews out to the other planets and the moon. Now, you may know the Starship, the first time it was tried a couple months ago, it did explode, so it's not ready to go up quite yet. It did have what was called a rapid unscheduled disassembly, which is what I call my Friday nights sometimes, basically. You know, it blew up. Sometimes you just collapse on a Friday night, but it's still, the fact that it flew was pretty exciting. That means this can get, the fact that you could decrease the cost almost 100-fold of the cost per kilogram for spaceflight crews and materials is really extraordinary. So it didn't quite make it yet, but it's on its way and it's very exciting. And finally, one of the things we're doing for the Polaris Dawn mission is taking the new nanopore sequencer, and this is the Mark 1C, which is actually made by Oxford Nanopore Technologies here in the UK, where actually it's a small sequencer. It's actually, you know, not that much bigger than this control. And actually, we're going to be flying this hopefully in flight to do drawing of blood and sequencing and epigenetics and also DNA damage measurements right at that moment in flight. So I want to close in the last few minutes of thinking about ending on more of a perspective note and even a philosophical note at the very end. So I want to go from what we've been doing and what we all do, I think, a lot, is we measure the effect of drugs, we measure the effect of stress, we measure biology as often and as comprehensively and as thoroughly as we can. But philosophically, I want to become eventually more active. Could we think about ways to have defensive biology in advance of a disease, in advance of a stress, or in advance of any sort of radiation, for example? And we know, for example, that even astronauts' urine shows damaged DNA during flight. This is from 59 crew members, where if you look at 8-oxoguanosine, or the measure of DNA damage basis of guanosine, you can see it spikes up in flight day 15, and eventually goes back to normal when they return back to Earth. But it's consistently showing damaged DNA being sort of eluted out of the bodies of these astronauts again and again and again. And that was actually, that's in relatively safe environments, right? So Scott Kelly, for example, when he was in space for that year, is 146 millisieverts of an effective dose of radiation. And the Inspiration 4 mission there, the other light blue bar, was actually 4.72 millisieverts. So not even that bad in terms of the amount of radiation, because they're still protected by the Van Allen belts. We're in the relatively safe harbor of low Earth orbit. If you think about going to Mars, there and back would take maybe 3 years, it would be 1,200 millisieverts. Or if you were a Hiroshima atomic bomb witness, that's like 200 millisieverts. But Mars, you can see, is far beyond any of these other prior missions, and would be a very hard trip. So the thing we've been working on in the lab is, could you genetically protect astronauts? And we've written about this as well as others. But then it raises large ethical questions, like should you do such a thing? Could you have sort of transient activation of different genes potentially? And this used to be very hypothetical and peculiar for me to talk about, but it's really changed in the past 2, 3, 4 years, because now there are hundreds, at this point actually now over 1,000 CAR T and CAR modified trials, chimeric antigen receptor trials, where we're modifying T cells. This is one of our clinical sites in Chengdu, and then infusing them back into patients. And so we now, like I said, there's actually over 1,000 of these different clinical trials for engineered cells being used therapeutically. And more impressively, I think in the past couple of years, there's also been some successes for transfusion-dependent beta thalassemia or sickle cell disease. We're doing CRISPR of the patients, so somatic CRISPRing, basically in this case, turning back on fetal hemoglobin in the body of the patient. So if you have a problem with your adult hemoglobin or alpha and beta, why not turn back on your fetal hemoglobin, which you used to use when you were a fetus? You might not remember it, but you had that gene inside of you that was active once, and you can turn it back on. In this case, it's a curative treatment for sickle cell disease, and it's extraordinary to see this idea of a somatic CRISPR being used therapeutically. And that's really if you want to modify the DNA. But if you think about it, there's other ways and tools you can tweak genes. In particular, you can use on the fabric of life, you can actually change epigenetic states, and instead of just adding or deleting or modifying DNA, just change how and when genes are activated. So it's actually a great review paper from Charlie Gersbach that showed many applications in many ways. You basically can take a deactivated Cas9, the enzyme that guides CRISPR activity, and then link it to, say, TET1 or DNA methyltransferase, like DNP3A, and go in and turn on or off specific genes or modify histones or DNA to turn on target genes when you want them to go on. And so what we've been doing in the lab in the past year and a half is actually doing just that, is doing targeted deactivation or activation of space-related stress genes. Now, this is just in hex cells, just in vitro in our lab, but it shows that you can actually take some of these constructs. We've been trying 3 of them here, and you can see either turn down specific genes or turn on sometimes 50 or 60-fold specific targeted genes to flip them on like a light switch. And so you could imagine you have genes just turn on during a high radiation environment and actually keep you safe as needed at very specific times. And so that's one thing we've been doing. The other thing we've been doing is actually thinking about other creatures that can survive the ravages of space, like tardigrades here. These cute little water birds can survive in the vacuum of space and be rehydrated and brought back to start walking around. And this actually, when their genome was actually published, the Hashimoto paper in 2016, they found this protein called DSUP that they estimated could give a 40% reduction in DNA damage with just a transient, basically, expression of this DSUP protein. And so we've taken inspiration from this paper and as in follow-up work to say, well, could we get it to be stably transduced into human cells and actually have human cells making DSUP constitutively? And indeed we can, and they actually will go to the sites of double-strand DNA breaks and move in and help repair essentially the damage quicker. And in this case, I'm going to go back here and actually make it so they can get an 80% reduction of that DNA damage, what we're seeing so far. So it can take lessons from potentially any creature and use it to help prevent the damage or repair that damage faster. And so the other thing we're thinking about is, could we, take these kind of chimeric creatures and have it make it so you get them on Mars, that maybe something could grow on Mars, or have these hybrid Arabidopsis that could essentially survive in the Martian environment. So it would take a long time, probably dozens, hundreds of years to get there, but we've put a roadmap here of ways you could actually get to Mars essentially sustainably and make on-demand pharmaceuticals and therapies on that planet rather than relying on everything coming from Earth. So it'll take a long time, but I'll come back to this at the end of the talk. And the idea is, could you make plants and animals that could survive on Mars? And so one of the other things we're doing in lab is trying to find other creatures that could help us make that trip. So in particular, one of the projects is called the Two Frontiers Group. This is, they're out looking in places where there's high and large amounts of carbon sinks, where there's essentially excessive carbon dioxide. Take those creatures that are surviving there and actually learn from them. And so far we just found one cyanobacteria that actually has, it can eat carbon dioxide faster than any other cyanobacteria that's been found so far. So we're thinking about ways you can, again, learn from other creatures who've adapted on Earth and begin to use them to think about long-term applications on Mars. And I want to close in the last few minutes on the philosophical endpoint. It's just why. A lot of people say, well, why work on spaceflight research or these microbes that could survive on Mars when there's poverty and other issues on Earth, there's other diseases, there's other problems that are pressing, that are here right now. And I think the big answer to why do this kind of research, I put into 3 parts. The first response is that we can address serious diseases and problems here on Earth while advancing science and medicine in space. You can actually walk and chew gum at the same time. We passed civil rights legislation in the United States there in the 60s and got people to the moon. We know we can do both things at the same time. The second reason is I think it is an intangible but essential asset to have hope, to look up at the sky at night and know that there's people imagining lunar habitats, that people are literally reaching for the stars, and the next generation is something that they can look forward to, is a very hopeful and essential part, I think, of humanity. And the third reason really is, I think, why I do this kind of research is because it is actually, I argue, an essential duty for humanity, and really just to preserve life, because life is precious. The third reason is that life is not guaranteed. Even in North America, about 1/4 of all birds have disappeared over the past few decades, mostly through habitat destruction. A lot of microbes have also been lost before we can even sequence them or find them. So this is the idea of our modern lifestyle leading to a lot of health problems, as well as just losing species before we can even find them as part of this sort of mass extinction that we're worried about. And this is, not the first time. If you take the past 500 million years of our planet and look at what's happened, the temperature, of course, has gone up and down for the past 500 million years. You can see in orange, we've had a world without polar ice caps. We've often had a world with polar ice caps. You can see right now we're in a world that does have polar caps. but it hasn't always been this way. But anytime there's been a large, rapid change in the temperature of the planet is when we've seen a mass extinction. So far, 5 of them have occurred. The big concern now is, of course, if we increase the temperature too fast, we would create a 6th mass extinction. Some argue that's already happening. And so this is, just terrifying to look at, but it's actually, this 500 million years, if you zoom out a little bit more, you can see these are the major extinctions, but zoom out a little bit more. There was a time 800 million years ago, in what used to be a snowball Earth. It was much colder. It feels like it was cold outside today, but it was much colder not that long ago, 700 million years ago. And if you take this Earth and put it in the history and likely future of our planet, this 500 million years kind of sits right in the middle of this very, where there's liquid water on our planet, and the Earth formed about 4.5 billion years ago. And we're in this very nice but temporary temperature canyon of our planet, where eventually actually the sun will start to increase its luminosity. It'll start to boil the oceans. When I was a kid, I always thought that we had like 4.5 billion years until something really bad would happen, like the Earth would be engulfed by the sun. We really probably couldn't survive then, but actually it's only about 1 billion years until the sun will boil off the oceans. It's going to be pretty hard to survive then. So I was finishing a book about this, and I went down to my kitchen, and I was kind of moping around my kitchen, and my wife was saying, "What's wrong with you? What's happening?" And I just said, "I just thought we had more time." Because it was 1 billion years, but that was much less than 4.5 billion years, which I had always thought. Imagine if you were going to be 100 years old, and then someone said you're going to be dead by 20. Of course you'd be upset. So if you had a time-independent view of life, and if you cared about all of life, then you would be upset by this, I think. So I keep thinking about this plot, and I keep thinking about all the life that's in jeopardy, right? So there's producers, consumers, decomposers. This is what we've always been taught. And there's 3 kinds of life. But really, this is, I think, missing something in this picture. There's actually a 4th kind of life. There's the only kind of life that's aware of the frailty of the ecosystem or of life itself. Quite literally, only humans can be sort of the guardians of this life, of these life forms. Quite literally the guardians of the galaxy, if you will. I think it argues what we are, because we're the only ones that know that life is at risk, that only humans are extinction aware, and therefore only humans can actually address it. We are the only ones that can actually exercise this duty to all life, present and future, maybe even in the past. And we have done it imperfectly. We've caused extinctions, for example, for the dodo and other species. But there's a chance maybe now to also use technology to maybe keep species alive or even de-extinct species. This is one example, like the black-footed ferret, which used to be all over the western United States, but now is slowly being reintroduced. And in this case, using cloning technology to actually take some of the DNA and put them into donor-gestated ferrets. And you can see here one of them was born, Elizabeth Ann, who we just were sequencing in our lab, is to see how well we can do this epigenetic reprogramming to keep species alive in the next generation. And I think this is an exciting project, but it's not. There's also a company, full conflict of interest. I'm on their advisory board, but Colossal is trying to bring back the woolly mammoth, as well as other species potentially. And this is, this idea of using technology to preserve and even bring back species, including the dodo, for example, is now on the list, and the company is working to bring back the dodo, which raises big questions, because if you take away a species from an ecosystem, it could be devastating. If you put one back in, or anyone in, it could also be devastating, or it could be helpful. So it's going to have to be, very careful and very slow. And there's a lot of plans already in the works to make this possible, which I think is fascinating. And so finally, I think this is a duty that we have towards all life, past, present, and future, and something I'm going to shamelessly plug in a book that I just finished and published. It includes the story of me being sad in my kitchen at the end of it, what I think we can and should do for the next 500 years, as well as some vignettes about the Scott Kelly mission and some of the work on the twin study. So with that, I want to thank, I think we're saving questions maybe for the end, but I want to thank everyone in the lab that makes this work possible. I could not stand here without having an extraordinary team of scientists and passionate researchers with whom I get the pleasure to work. And also, this is a more recent picture of some of the lab and everyone cheering when people could finally start to hang out again after COVID. They're very excited. And again, thanks to support from NASA, the NIH, World Quant, several other foundations, as well as support from SpaceX and Trish and Star Cancer Consortium, many friends, colleagues, and collaborators on the list, and gratitude to all of them. And thank you so much for your time, and I'll look forward to more questions later.
All right, Chris's talk is open for any questions.
How many of you want to go to space? We can do a raise of hands. If you ask an elementary school class, every single kid puts their hand up, I'm just telling you. Anybody worried about the woolly mammoth?
Hey, Chris, I was fascinated by the male-female difference. Do you know if, I would imagine there's an age difference as well. Is there a striking sort of cutoff that you see?
Great questions. The question was, we see sex differences, there are also differences by age. We looked actually at telomere length as a function of age, and that the older, there was no big association there. We thought, actually talking with Susan, I know we're going to play her talk next, is that was not a biggest factor. But for gene expression, we actually, we see that the younger astronauts do seem to have a little bit more plasticity. They return to baseline a little bit faster. It's a small cohort, so I can't say definitively, but I think as we start to get the Axiom data, other data from Polaris Dawn, other missions, or even as we were talking about before the session, if we just have more and more controlled data sets, I'd be fascinated just to see how often you just take 500 normal people and look over time, and you do any kind of perturbation of any kind, how fast do the young people revert back versus the older people. The presumption is that as you age, you get immunosenescence, the cells are less plastic in general, and so that's what we're expecting to see for other cohorts.
Very good. Well, if there's no question,
Maybe one there.
Yeah.
Thanks. That was a nice and wonderful presentation. I just want to know,
Thank you.
in an experiment where you determined that DNA damage in people in flight. So I just want to know, where do you take the sample? You take the sample when they are back on Earth, or you take the sample while they are in space? And if you take it in space, how do you make sure the sample, when taken back on Earth, is not affected by environmental factors on Earth's surface? Thank you.
The fabulous question. That's one of the biggest confounding variables for any study, but especially spaceflight studies. So whenever possible, we use the same tubes, the same centrifuge, the same protocols in flight, and then freeze the sample. There's a freezer on the space station, the current space station, and there's also, for some of the Dragon capsules, though, they did not have cold storage, so we just did a Wattman card for the blood draw and just blood spots. But for future missions, we're doing actually the blood collection and sequencing within a matter of an hour. So we'll try and then do the same protocol before and after flight. So basically, whenever you can, you match the protocol exactly in flight or before or after flight. And you also use the same tube, same lot number, same protocols. The other thing we do is often compare whenever we can to other controls, different cohorts, things like the UK Biobank, the Iceland genomic study, Ireland has a genomic study as well with RNA sequencing. So we always try to put into context what is baseline variation from just people experiencing stress of any kind, and subtract that as part of a linear model. We use generalized linear models to put that into what is the framework of noise and accommodate that as best we can. But that is one of the hardest things of getting samples in space, is you basically want to draw the blood and immediately put it in the freezer right after you spin it, so at least it stays at -80. There were some examples from the twin study, which actually you'll see in the next talk. We got blood samples that were drawn in space, put in the Soyuz, and brought back to Earth, and then shipped to Houston. And samples got repatriated within about 36 hours. But that means you then had a tube of blood that was moving around the planet, and then in a plane, and then we got to the lab, and we did assays on it. But to simulate the difference, we took some samples of blood and put them on FedEx to go around the country for 36 hours, and then did the same thing. So whenever possible, we try to simulate any disturbance of that sample, if there is one.
All right, good. We'll move on to our next speaker.
All right, thanks.
The next speaker is Susan Bailey from University of Colorado. Susan could not join us in person, so she's actually recorded her presentation, which we'll play, and then she will be on live via Zoom to take any questions at the end of her talk.
Good afternoon, everybody.
Well, thank you. I really do appreciate the opportunity to speak to you today, and it's a pleasure to be here. And it's always a lot of fun to talk about the twin study, and I'll be reviewing some of our findings from that study, as well as highlighting some of our newer studies, as well as potential mechanistic insight that we've been gathering over the years since the one-year mission in the twin study ended. Primarily, it involves, mechanistically, it involves chronic exposure to the space radiation environment and how that can underlie some of the changes that we see in telomere length dynamics, as well as the persistent DNA damage responses that we observed and that were associated with space flight. It's interesting that the International Space Station has supported a continued human presence really for more than 20 years. The space station orbits in what they refer to as low Earth orbit, or LEO. It travels at about 250 miles above the Earth's surface at a speed of about 17,000 miles per hour. It also represents, of course, a very unique lifestyle associated even with that type of space travel. And it has extraordinary challenges and stresses. It also affords just an amazing view of our own home planet, Earth. And here you see a picture of the Northern Lights taken from space by Scott Kelly. So what I'd really like to do to start with here is to go ahead and highlight some of our findings, as well as here introduce some of the stars of the story that I'll be telling today. And you see here Kjell Lindgren on the front row on the left there. He's a NASA astronaut. And behind him is Kimia Yui, a JAXA astronaut. And over on the right-hand side, you see someone you probably recognize as Scott Kelly. And he was indeed selected as NASA's first one-year mission astronaut, which that mission was flown in 2015 to 2016, and certainly received a lot of press and very positive and exciting PR related to interest in space. We also probably heard some fake news that it was reported that Scott returned as a space mutant, which of course was not true. Chris Mason may be telling you a little bit more about that. But in fact, there was really a lot of real science that was associated with the one-year mission as well. There were lots of samples and studies for the whole year before the one-year mission, as well as the 1 year of the mission, and then for up to a year following the mission. But most of the PR and attention around the one-year mission was really more related to the fact that Scott Kelly had an identical twin brother, Mark Kelly, so who had also been a former astronaut and Navy test pilot. So you had two very similar individuals, similar in nature and nurture. And they asked the question at the press conference, are you doing anything? Because we have, I have this identical twin brother. So it didn't take long to realize that sitting right in front of them was the perfect experiment, that they had one twin spending a year in space and his identical twin ground control spending that year on Earth. And that's really how the twin study was conceived and born. And it really was the first time that NASA ventured into what we might consider 20th century omics, or really getting into the field of omics, again, more of which Chris Mason will be telling you about. And they selected 10 studies, 10 investigations from around the country that covered all kinds of different pieces of omics, as well as physiology and, of course, telomeres, which I'll be talking some more about. And I would like to just kind of start by highlighting a few of the other firsts that were involved in the twin study, and some of the important findings from that, to just give you a feel for how comprehensive the study really was. First of all, if you start thinking about even molecular studies at all, one of the first things you have to be able to do is pipette, and thinking about doing that in space is a bit of a challenge. If you think about it, microgravity, you've got tips flying all over the place, and how do you manage, liquid handling in space? And Andy Feinberg and his team at Johns Hopkins were really instrumental in pioneering some of the methods used to be able to do things like simple things, like even as easy as pipetting here on Earth. And in fact, that led the way to the first DNA sequencing that occurred in space, which was done by Kate Rubins in August of 2016. And really now it's almost routine in space to do sequencing, or at least as routine as anything is in space. Chris Mason, who I've already mentioned a couple of times and who you will also be hearing much more about, and his team were also very interested in DNA and RNA sequencing, as well as methylation, so epigenomics, transcriptomics. So again, you'll hear a lot more about the omics approaches that he was very much involved in pioneering as well. Scott Smith and his team at NASA do biochemical profiling on the astronauts, so blood and urine collections, and monitoring a lot of different metabolites and different markers in the blood. And of course, one of the important things to think about is diet when for the astronauts, and that's because we are what we eat, and the diets on station are very defined and very limited. So even though they might be very healthy, they're also kind of boring. So the astronauts do, excuse me, struggle with that just a little bit. Okay, so you see Scott Kelly and Kjell Lindgren actually eating the first space salad. It was lettuce that was grown on station. And when you think about it, I mean, it's really important to be able to start as we travel further and further in space and stay longer periods of time. It's going to be more and more important that we're able to grow crops, grow plants, and they also have to be able to survive the mission. So a very important area as well. Here, Emmanuel Mignot also did some immune studies, and here the important thing was actually testing a vaccine in space, which hadn't been done before. So here you see both Scott and Mark Kelly giving themselves a flu vaccine. And in fact, the response in both was very similar. So that was encouraging that our vaccines will work during spaceflight. Here is Stuart Lee, also at NASA, involved in some metabolomic studies looking at cardiovascular effects. And Brinda Rana was also involved in some of these studies. And you'll hear more about that as well as some of the work that she's also done with proteomic assessment of fluid shifts. So one of the things that happens when crew members get into microgravity, into space, is that the fluids that usually collect more around our legs because of gravity actually float up, and they don't have that anymore. So there is increased pressure and fluids around in the head, and oftentimes astronauts will have problems with vision or headaches or increased blood pressure because of these fluid shifts. So you can see some of the instrumentation here that they have to measure that during spaceflight. Cognitive performance is also another one that was evaluated not only in the twins, but fairly routinely in the crew members. And this particular study was done by Matthias Basner at University of Pennsylvania. And again, they are cognition tests that are computer-based, so a battery of tests on the computer that they take before, during, and after spaceflight. And in fact, Scott Kelly did have some decline in cognitive function post-flight, and it took him 6 to 9 months to really recover from that and feel more like himself. If any of you are interested, Scott Kelly wrote a very interesting book, an autobiography called Endurance, and he talks about a lot of his experiences, including that one. The first time that the microbiome has been evaluated during spaceflight was also done as part of the twin study by Fred Turek at Northwestern and his team. And again, as you would expect, yes, it definitely changed during spaceflight, but then it also recovered very quickly upon return to Earth. So I hope that very whirlwind tour through the twin study gave you at least a small appreciation for how comprehensive it was, and also really how it was stood at the forefront of personalized space medicine to evaluate the health effects of spaceflight in individual astronauts. And there's certainly little question or no doubt about the fact that this pair of twins is the most profiled on or off the planet. It's also true that they're one of the most interviewed because they just had so many interviews around the twin study, generating so much interest and excitement about space travel. And it's interesting that one of the most common questions asked of Scott Kelly was whether or not he would return younger than his brother Mark because of his spaceflight. And it was usually asked in the context of Einstein's twin paradox or perhaps of watching Interstellar. but really and truly those who have done those calculations assure us that even if there was some time dilation, it would only be a few milliseconds because of the the space station is traveling, is orbiting the Earth and not traveling at the speed of light away from us. So it really would be very minimal, but still the question of aging and perhaps accelerated aging and increased risk of developing age-related pathologies like dementias or cardiovascular disease, and even some cancers like leukemia that could come up within a time span of a number of years. These are things that could really influence astronaut performance during a mission, and they could also be very important for long-term health implications. So that is really where we come into the twin study, because we were the first to evaluate an actual biological marker of aging, as we propose that telomere length dynamics, or these changes over time in telomere length, would really serve as a very informative biomarker of general health as well as aging and disease risk. And that's because telomere length dynamics really does integrate all of the different exposures, extreme exposures, as well as the many unique stressors and challenges and experiences that the astronauts undergo while they're during space flight. So things like microgravity, for example, space radiation exposure, isolation, that very defined and limited diet, all of these are captured by changes in telomere length over time. And here you see Kjell Lengren on the right. He was in the previous slide as well, flying the Colorado flag in the cupola of the ISS. Here he is on his second ISS mission, second 6-month mission, commanding the Crew 4 on the SpaceX vehicle. So he just recently returned and has accumulated over 300 days in space now. So he's getting very close to as many days in space as Scott Kelly had as well. So I've already talked about telomeres some, and I haven't really told you what they are and why they're important. Telomeres are just the natural ends of our chromosomes, and they shorten with cell division. They can't replicate to the very end of a DNA molecule, so they shorten. And so they also shorten with age. And that's why they've been referred to or regarded as a mitotic clock. But even more importantly, they're really becoming established as a hallmark and even a driver of aging in humans. And in addition to cell division, things like inflammation and oxidative stress and a whole variety of lifestyle factors, including nutritional and psychological and physical stresses, as well as environmental exposures like UV and ionizing radiations, all contribute to telomere erosion. And it really is this idea of how quickly or how well one is aging is related to how quickly our telomeres may be shortening, and the many different experiences and exposures that we have that might accelerate that process. I'll also just mention that telomerase is the enzyme, a very specialized reverse transcriptase, that is capable of adding telomere sequence to newly replicated telomeres, but it is only sufficient to maintain telomere length in the germline and stem and cancer cells. So that is why in the majority of our cells, telomeres shorten with age. Again, I just stressed this idea of telomere length dynamics, or changes in telomere length over time, is really what's important when we think about a biomarker of aging or of the associated age-related diseases that go along with aging. Things like reduced immune function, loss of fertility, dementias, cardiovascular disease, and cancer. And then telomere length dynamics then really integrates and captures this interplay between genetics, life experiences, and exposures, and can be viewed somewhat as a sentinel of these kinds of exposures and be a biomarker then of general health and aging and disease risk, because it captures all of these different things, including space radiation, which again is one of the major hazards associated with long duration space flight. And that is because, you know, the Earth offers us quite a bit of protection from the more damaging types of radiations because of our atmosphere and magnetic field. They really do shield us from some of the more damaging types of radiation. So once you're outside of that, that protection, the doses are very much higher. And so you say, well, maybe shielding is the answer. Well, the problem with shielding is, of course, is that it's very limited, and that's because it's really usually very heavy. Things like water or concrete are, you just can't you know, fly those. So shielding is a problem, and sometimes it's non-existent when you think about spacewalks or EVAs. So, and in some instances, shielding is even makes things worse because of secondary particles like neutrons. So as people, as we journey on back to the moon as well as onto Mars, these things are going to, radiation exposure is definitely going to be more and more of a concern. Doses will be higher, and astronauts will be exposed to more and more damaging types of radiation. Just to give you kind of an idea of the doses, a 6-month mission on the ISS, doses are estimated around 100 millisieverts, and that's compared to average natural background, which would be around 1 to 2 millisieverts. They estimate that a 3-year journey to Mars would be more like around 1,000 millisieverts. So again, quite an order of magnitude higher than what a 6-month mission on the ISS is, just to kind of put that in perspective. And it's also interesting to think about the only time that we've actually been outside, completely outside of the protection of the Earth, was on the Apollo missions. And here you see a very interesting and illustrative film that was taped to the ankle of Neil Armstrong on his 8-day mission on Apollo 11 when he landed on the moon. And here you see some different tracks of ionizing radiation. You see kind of sparsely ionizing gamma rays and X-rays that are these little blobby kind of dots scattered throughout here. You also see some tracks of protons. These kind of lighter lines would be protons. And then you also have this really densely ionizing high Z or HCE particle that's more like a train track going through the cell. So even after 8 days, you know, you can get an appreciation for the different types of ionizing radiation that astronauts are exposed to. So all of this really is, I've told you this and really made the case that it led us to hypothesize that surely because of all of these really extreme environmental exposures and really unique challenges and stresses, that spaceflight would surely act to accelerate telomere shortening, that they would experience telomere shortening because of spaceflight. And so we were very fortunate, of course, to be part of the twin study where we had the twins and we could evaluate telomere length as well as telomerase before, during, and after spaceflight. And we could make comparisons between the twins who were genetically matched, so we could identify spaceflight specific factors that might influence telomere length. But we also had a separate cohort of 10 unrelated astronauts, and as well as their age and sex matched controls. And we could use those as well and make comparisons that would help to allow us to evaluate the role of not only genetics, but also individual differences and susceptibilities between individuals. I've also mentioned a number of times too the importance of this longitudinal nature of the study, that in fact we were able to get samples both before, during, and after spaceflight, Here you see the actual collection dates that we had. And so really very straightforward in design, but much more difficult in execution and actually getting some of these samples. You can appreciate that astronauts are very busy and they're all around the world with their training and other responsibilities that they have. So just catching up with them, it can be difficult. But also the in-flight samples were very, very challenging. And that is because we needed ambient blood. So what they would do, and you see in the middle here, a couple of the astronauts, Tim Peake and Tim Kopra, doing a blood draw on station. And they would do that right before the Soyuz vehicle returned some of the crew to Kazakhstan. And so they would send the samples down on the Soyuz, and then they would also go ahead and jet them back to Johnson Space Center with the NASA astronauts. So we were able to get our samples in the lab within about 48 hours of draw on the station, which is really quite remarkable. So we'll go ahead and let's dive into some of the data. Once we had those samples, they were processed for telomere length, both by isolating DNA and looking by qPCR at telomere length. We also did, we prepared metaphase chromosomes for telomere length analysis and chromosome analysis. Here what we're looking at is the qPCR data for telomere length on the twins. And as we go through this, you'll see that we have Mark Kelly in green, so he is our ground control, and Scott Kelly in blue, he's our space twin. And we've also averaged all the telomere length measurements for pre-flight, mid-flight, and post-flight. And you can see here that if you look across, well, first, let's just look at Scott and Mark, both starting off at relatively similar telomere lengths, which would be expected because of the genetic component of telomere length. And then you can also see that Mark Kelly in green has his telomere length is very stable throughout the study, so it doesn't change very much. But the big surprise came when we looked at Scott Kelly, as we see here his pre-flight, and then during space flight, instead of going down like we thought, it did exactly the opposite. And in fact, average telomere length was significantly longer during space flight than it was pre-flight. And then it went down again, it decreased again after space flight, and recovered really over the 9 months that we got samples from him. It recovered pretty much to his pre-flight levels. So that was really quite a surprise to us. We were very anxious then to see if we saw that in the other astronauts as well. And you're seeing that data here. And here we have all of our ground-based controls, everything normalized to the controls. We have the pre-flights for all of the astronauts here, and then we see, indeed, mid-flight, we see this significant increase in telomere length during space flight, and then a significant decrease in telomere length post-flight. So it was really very informative and important that we had these additional samples that we could start really attaching some significance to these findings. And in fact, we were seeing it consistently in our in-flight samples. We also, to validate those qPCR measurements, we also did telomere FISH, or fluorescence in situ hybridization, which is a cell-by-cell assay where we look at metaphase chromosomes and really analyze thousands of individual telomeres. And we do that simply by measuring the intensity of the fluorescent signal. So the brighter the signal, the longer the telomere. And it also allows us to evaluate genome stability as well, which I'll get back to here in just a minute. So when we do the telomere FISH, or the telo FISH, here's the twins data. Again, you can see very similar trends as we saw with the PCR. Mark Kelly not changing a lot. You see some decline in the post-flight samples. But again, we see this increase in telomere length in Scott Kelly associated with space flight. And again, with the telo FISH, with the entire astronaut cohort, we again see the same thing, and it's starting to actually be able to attach significance to it. In fact, pre-flight samples from the astronauts really were in general shorter than our controls, and mid-flight they were longer, significantly longer, and then post-flight they were again shorter and shorter than what they started with for sure. One of the really great and important things that we can do with the fish data is generate these telomere length distributions. So across the top, you see Mark Kelly, and then on the bottom, we see Scott Kelly. And again, if you look across here for Mark Kelly, you know, we look from left to right, short telomeres to long telomeres, and then also just the number of individual telomere counts that are here: short in yellow, mid-range in blue, and long in red. And you can see that these distributions are fairly stable over the course of the study in Mark, and that was just, you know, really very supportive of the average telomere length measurements that we were getting with the qPCR. So very consistent with that. A very different story when we look at the bottom panel here and look at Scott Kelly. And we see again that he's got a very different distribution of short and long telomeres that changes quite a bit during space flight in the middle here, that in fact we're seeing a reduced number of short telomeres and more long telomeres, which again would be very consistent with the increase in average telomere length that we observed during space flight. And then look what happens post-flight over here on the right. You see again another dramatic shift in the populations of short and long telomeres, where we see an increased number of short telomeres and a decreased number of long telomeres. And this is really where we feel like the long-term health implications is that in fact he now has many more short telomeres than he did before he started. In case I haven't convinced you yet that this is real, again, in collaboration with Chris Mason and his team, we were able to also look at sequencing, telo sequencing. So they did the sequencing of telomeres. And here you can look at telomere length, and you see here is Mark Kelly on the right. And again, very, very much the same throughout the study, before, during, and after spaceflight. And then over on the left, we have Scott Kelly. And again, you can see this increase in telomere length mid-flight, and then a return to closer to normal. So we even saw it in the sequencing. Another twin investigator, Kumar Sharma, looked at telomere length in urine, and he did this with qPCR. And again, we see exactly the same thing compared to Mark Kelly. Scott Kelly had this significant increase in telomere length during spaceflight. So by all measures, and really quite independent of mission duration, because most of the other astronauts were on about 6-month missions, and just regardless of how we looked at it, it was a very consistent phenotype associated with spaceflight. And this is just really hot off the press. And again, in collaboration with Chris Mason, we looked at the SpaceX Inspiration 4 all civilian crew, who were only on a 3-day mission, a little more high elevation than the space station. But even after 3 days, we see that in fact telomere length is elevated mid-flight for that crew as well. So that is really telling us that it's a very rapid response, and it also seems to be a very consistent response to spaceflight. The other thing that we've been doing with Chris Mason is looking at telomeric RNA. So telomeres are actually transcribed into RNA, which is called TERA. And we started to look into the role of TERA, you know, what was it doing? And asked the question about what happens to TERA in response to spaceflight. And in fact, looking both at the Inspiration 4 crew, we see this increase. The red, orangey red color is more of an increase in TERA transcripts. And you can see that in fact, during spaceflight, even 3 days, they did have elevated levels of TERA transcripts. And going back to the NASA twin study, we see something very similar. in Scott Kelly during the 1 year mission that in fact tera levels were increased. So this is a very exciting finding that we've been trying to characterize with some of our in vitro studies. And in fact, it seems to be related to tera being involved in the DNA damage response and perhaps repair of damaged telomeres. And in particular, it seems to be responding to double strand breaks. And the reason that is important is that ionizing radiation is a very potent inducer of DNA damage and specifically of double strand breaks, which double strand breaks, of course, just break both strands of the DNA, and so they break chromosomes. And one of the real signatures of ionizing radiation exposure are chromosome aberrations or rearrangements, because sometimes these broken ends can get back together incorrectly. And sometimes that occurs between chromosomes, so we can pick up, here we're using directional genomic hybridization with probes to 3 of the different chromosomes, and they're all labeled with red. You can see them as red here. And we pick up translocations, as you see here, where there's the signal actually is split now between 2 different chromosomes, so you have a rearrangement between chromosomes. But we can also pick up rearrangements within chromosomes. So here we see an inversion, and that just means that a piece of the chromosome broke out and it popped out, it inverted and went back into the chromosome. And that process then makes that signal, the red signal, hop over to the other side. We're currently able to do all of the chromosomes. We can do them in different colors and actually do the karyotype so we can identify what each chromosome is, as you see here. And that's really been very powerful and really makes it much, much easier to do this analysis for not only translocations, but here you see a very nice example of an inversion on chromosome 1, where we just have that block of signal hopping over to the other side. And again, both of these rearrangements, translocations, and inversions are very characteristic of ionizing radiation exposure. Here, let's look at the bottom panel, and we see when we look at the entire cohort of astronauts, we see that indeed a space flight increase in inversion frequency that actually persists. It didn't really go down during the course of the study. Point out that dicentrics are also an important marker of radiation exposure, and we did see a few of those, but again, inversions were really helpful in that we saw higher frequencies of those, so didn't have to score near as many cells. And it was a very consistent finding across the astronauts. The other thing that we found that was very characteristic of spaceflight and very specific for it were these satellite associations that you see here. And those are just where the short arms of the chromosomes essentially associate with one another. And they're important because they've been reported before in occupationally exposed hospital workers. So they can serve as markers, biomarkers of chronic low dose kinds of radiation exposures. Across the top panel here, you see too that we also looked at what they call sister telomere exchanges, just another type of exchange that happens within chromosomes, and really are characteristic of some of the higher LET or densely ionizing radiations, as you would expect in space. And again, we do see some space flight specific increases in those kinds of events, which again is just very supportive of the type of exposures they're getting. If we think about the telomeres over here on the right-hand side, this hetero sister telomeres just refers to heterogeneous telomere lengths on the 2 sisters, and I'll show you a picture of that in a moment. But you can see again that it was very spaceflight specific increase in this rather unusual telomere phenotype. And this is actually what it looks like. You can very readily tell how heterogeneous or how unequal the 2 telomeres are on this particular chromosome. So one's very large and one's very small. And that is actually considered a hallmark of what they refer to as ALT, or alternative lengthening of telomeres. It's just a telomere length maintenance pathway that does not require telomerase. And it can be transiently activated with high LAT exposures or with chronic oxidative stress. Again, both things that you would anticipate and expect to be associated with space flight. The satellite associations that I mentioned, here's a picture of those, and you can just see those little short arms associating with one another. And that has been previously proposed as a biomarker of chronic low dose radiation exposure, which again would be very consistent with spaceflight. So I'll just, to kind of summarize what I've been telling you, and proposing that really telomeres are very robust biomarkers of different effects associated with spaceflight. For the twin study, we mentioned that we saw, in fact, that the twins started the study with very similar telomere lengths. But in general, astronauts really did tend to have shorter telomeres than our healthy age and sex-matched ground controls before flight. So a lot of that could be, the many stresses that they're under prior to flight, training, and all the rest that goes along with that. I didn't really go into any of the telomerase activity data, and that's primarily because of time, but also our in-flight telomerase activity assays were not successful, so we didn't have a direct measure of telomerase activity, but we did see evidence that certainly telomerase activity is influenced by life stress. And the most striking finding, and really the most consistent finding now that we've observed, is the significantly longer telomeres during spaceflight. And that was in all the crew members for whom we had in-flight samples, and then for all the in-flight samples that we looked at, that was true. It was independent of mission duration, whether it was 1 year, 6 months, or as I mentioned in the Inspiration 4 crew, only 3 days. And it was also independent of the means of measurement. We saw it with qPCR, the telofish, as well as the telomere sequencing. And then I also mentioned that our newest finding is related to telomeric RNA, or TERA, and its potential role in repairing some of the damaged telomeres. Telomere length also shortened very rapidly upon return to Earth. And for Scott Kelly, we had samples within like 48 hours, and they shortened very, very rapidly. And then over the course of the following months, they did tend to recover. But overall, the astronauts really did end up having more short telomeres than they started with. And as I mentioned, that's really where we think the long-term health and aging implications may be, and where, some kind of countermeasure or intervention may be, necessary or called for when we think about the aging and age-related diseases that they would now be at increased risk for. The mechanistic insight that we have at this point really is related to chronic exposure to the space radiation environment. We see these persistent DNA damage responses during spaceflight. I mentioned the inversions and the satellite associations, which really are signatures of space radiation exposure. Chris will be talking more about the chronic oxidative stress and mitochondrial dysregulation observed during spaceflight. And these things also correlated with telomere length and were consistent with transient activation of the ALT pathway and these heterogeneous telomere lengths that we saw as well. So it's the story is starting, the pieces are starting to fit together. And even in another cohort that Chris and I collaborated on, of these climbers, twins that climb Mount Everest, a somewhat similar high altitude and extreme environment, we saw telomere elongation as well as this increased TERA levels. So we're very keen about following up on some of those studies as well. And again, I think it just really supports the consistency of this phenotype. It could be related to some of the telomere elongation and trying to answer the question of why. Again, I think thinking about lymphocytes being very radiosensitive, it makes sense that a lot of them are dying, and perhaps what we're really seeing is this redistribution of leukocyte subsets and looking at different cell populations, different cell dynamics. And that's why we're seeing some of the changes in telomere length over the course of the study. And so it may not really be that telomeres are actually growing or getting longer per se, but it may be a combination of some of these contributing factors like chronic oxidative stress, telomere damage triggering some of this transient activation of ALT, as well as the changing cell populations over time, and actually working to kill some of the lymphocytes, so you have increased cell killing as well. So again, I think radiation exposure is another example of an area where mitigation strategies are really called for, and not just for aging considerations, but just in general, a very important area to figure out how we might be able to better protect the astronauts or mitigate the effects of radiation exposure. So by this time too, you might be asking yourself, well, why should I care about any of this? And really, we all care about aging, especially as we get older, and really maintaining health span, not just lifespan. And although, Scott Kelly came back from a year in space, he was briefly taller, and he was perhaps a couple of milliseconds younger, and 15 pounds lighter. It's really doubtful that space is going to be a fountain of youth. And I think that's probably very much related to the fact that when they come back, that they have perhaps accelerated some of these processes and have many more short telomeres than they started with. But if we really think about aging in the larger context and kind of expand our vision, thinking about, future colonization of planets and all of that, space really can be viewed as the next frontier of aging studies and of potential breakthroughs, which really are going to be required if, in fact, future space exploration missions are going to be successful. And perhaps that was something that even Spock realized or appreciated with his live long and prosper. But it's going to be especially true as the number and diversity of space travelers and even space tourists increase over the coming years. There's going to be a much wider range of age as well as health status to consider as well. And we've already seen, as a civilian childhood cancer survivor, as the youngest astronaut as part of the Inspiration 4 mission. And then we've also seen Captain Kirk or William Shatner as the oldest astronaut going into space at the age of 90. So there's just a lot of things that we'll have to consider, and the more we understand about it, of course, the more we'll be able to do about it. And certainly it is very exciting times to be a part of the space program, and I'm immensely grateful for the opportunity, and thank NASA certainly for making these studies possible. I also like to thank the people that actually did the work that I showed you today, those in my laboratory at Colorado State University, the graduate students, and Lynn Taylor and Carrie George have been really key in the assays that I've described. And certainly collaborating with Chris Mason has been really important and instrumental with integrating some of our results with some of the more omics-based types of studies. So a real thank you and shout out to him for that. And it's been really a privilege and a pleasure to serve with all of the twin studies investigators. It really was a remarkable experience, and I thank them all for that as well. And I invite all of you to really stay tuned and buckle up, because we are going and propose to return to the moon by the end of this decade. So with that, I thank you for your attention as well, and happy to take any questions.
Susan's joined us via Zoom for any questions you may have. Hey, Susan.
Hi.
Got one coming up there, yeah.
Yep.
Thank you for your interesting presentation. My name is Gerard Rongen from the Netherlands. I was wondering whether the storage conditions in the Soyuz could have affected your measurements. So did you have a control blood sample, for example, drawn on the ground, taken into the Soyuz and measuring it afterwards?
That is a very valid question, and it certainly is one that we feel like is the reason that we lost telomerase activity. I mentioned that our assays were unsuccessful for telomerase activity, and I believe it's because of the conditions on the Soyuz. We tried very hard to find out, what the temperature was, and they just, we really weren't able to get that information. The astronauts are in suits and things, so they even asking them, they weren't really aware of that. But I think time is also the other really important thing, and that is that we did have ground samples, blood, that was transported, and we did some experiments just transporting blood, with FedEx, on the ground. And if we could get the packages within 24 hours, we were able to get telomerase activity and all of our DNA isolation and telomere length measurements all worked just fine. If we went any longer than, if we went 48 hours or any longer than that, we certainly lost telomerase activity. But the good thing about that is that even at 48 hours, we were able to stimulate the lymphocytes, so we got metaphase chromosomes, and we were able to isolate DNA. So DNA, is really much more stable. And the fact that we could stimulate the lymphocytes gave us a lot of confidence that our telomere length measurements were valid. And again, we had so many different ways that we measured telomere length, but that's definitely why we lost telomerase activity.
Thank you.
Do you have a question, Chris?
One other question, good talk, Susan. Do you ever see something similar for some of those responses and inversions in, say, radiation workers or other control sets, and see how often they appear and how fast they go away?
So we haven't had any other. Well, I should take that back. We've done a lot of in vitro studies, with some of the space radiations at NSRL at Brookhaven National Lab in New York, where we definitely see increased levels of inversions with increasing LET. So the more damaging types of radiations, the more inversions we see. Those types of assays are very difficult, to do long term. Like there's not a follow-up for any of those in vitro ones. We have had we did a study on veterans, World War II veterans who had been exposed as weathermen. They missed the prediction of where the plume would go after a nuclear test. And so it went over the Marshall Islands and they were accidentally exposed. And when we got the samples from that cohort, it had been 6 decades later, and we still saw increased levels of inversions in the exposed versus our controls. So I think that's a really good example that yes, many of them are transmissible, and they stay in the bloodstream for long periods of time, and so they really are a very good biomarker, dosimeter that gives you an idea of exposure.
Great, thanks.
Go ahead.
Hi, Susan. Nice to see you.
Hi.
I was wondering if you have any plans to use some of the animals in the space station to look at different tissues.
Well, I've been so tempted by the mice, they do fly, mice, and mice have beautiful telomeres, but the trouble is that they also have telomerase activity in all of their tissues. So they're not a good model for human telomere biology because they don't really shorten. So we haven't done that. What we are doing next is I'm going to be part of a study that's flying plants, so Arabidopsis, and we're going to be doing telomere length measurements and telomerase activity in the plants as well, because as we go further and further, we're going to have to take plants with us too for food and just psychological benefits even. But so plants, definitely, yes.
All right, good. Any other questions? All right, thanks, Susan.
Thank you. I'm sorry I couldn't join you, but thank you.
Good to see you, thanks.
We'll move on to our final presentation for the symposia. It's Brinda Rana, my colleague from the University of California, San Diego.
Hi there. Thank you for inviting us to do this talk. Hemal and Hemal approached us a few years ago, I think, to do this session, and of course, because of COVID, it was canceled. But I'm glad the organizers still had interest in our work. So pleasure to be here, and I just want to mention two things that I'm a little self-conscious about. I've been here in Scotland. This is my 5th day, and my luggage still hasn't arrived. So therefore, the casual appearance, and I'm slurring a little bit, and not because I was drinking whiskey with Chris or anything. I had surgery on my head neck area, so I'm a little stiff. So as my colleagues Chris and Susan talked about, NASA is gearing for its mission to Mars. It keeps getting pushed back. When Scott Kelly went up for his 1-year mission, he was the first US astronaut who had spent a year in space. Previous missions were all 6 months or shorter. So that gave NASA an opportunity to study physiology, molecules, what happens in space. A mission to Mars, as Chris mentioned, will be a 3-year round trip. So NASA has a lot of work to do to figure out lots of questions on what happens to the human body during space travel. Is anybody in the audience involved with space research? Nobody? Okay. Well, I highly recommend, if you ever have the opportunity, either with the European space agencies or NASA, take it. It's a blast, right? So for future long-duration explorations leaving low Earth orbit, NASA has great acronyms. One of them is LEO. NASA needs to develop in-flight health monitoring methods, diagnosis, and treatment strategies. Personalized medicine approaches are a good way to predict what supplies will be needed to maintain crew health on a long space exploration like that to Mars, because they can't stop anywhere. They can't do what I did, like go to Costco Aberdeen to resupply when your luggage doesn't get to you. By the way, anybody who is from the US and goes to a Costco and they'll see an ATM machine and it says no fee, and you get excited, you're like, oh, here's an American machine with an ATM card, thing. Don't do it. It ate my card, so now I don't have luggage or cash. Okay, so what do astronauts experience in space? Chris and Susan talked a lot about it. Well, you've seen the movies too, so pretty much you kind of know. Exposure to radiation. Going an exploration trip to Mars will be leaving Earth's magnetosphere, so there'll be new cosmic rays that have not been experienced before by our astronauts. And as NASA officials say, it's not what they know that worries them, it's what they don't know that worries them. Microgravity, weightlessness, I'll be talking a little bit about that. Disruptions of the circadian rhythm, isolation, dietary limitations. This is a big area of research for Scott Smith and Sarah Zwart, who I'll talk about, their work I'll talk about a little bit. And carbon dioxide, I was really hoping that Chris would show this movie about the astronaut demonstrating fruit punch and carbon dioxide, but you can watch the movie online. Okay, so how does the body respond to the space environment? Every astronaut that goes up into space, the immediate consequence is hypervolemia, a drop in plasma, a drop in red blood cells, drop in blood volume. There's evidence for hemolysis as well. I've put some reviews along the way for those of you who are interested in kind of learning more about space research. Another thing that all astronauts experience is fluid shifts due to microgravity. Humans have evolved on Earth with 1g gravity. And so when you're on Earth, the gravity is pulling the fluids in your body towards your feet. We've learned to compensate for this gravity, this pulling, by pumping our fluids upwards so that we can feed our cells up in our brains, in our heart area. When we leave 1g gravity and we're in microgravity or zero gravity, the pumping continues, but there's nothing to pull the fluids back to the feet. So all the fluids start moving upwards and staying there. So if you want to imagine how this feels to an astronaut, just remember back to your childhood when you hung upside down on monkey bars and that pressure you feel in your head. Now imagine that for a year or 3 years when you're going to Mars. So this causes a lot of issues. One of them is facial puffiness. You know, when I was younger and I saw pictures of astronauts, I just thought it was some weird camera thing going on and made their faces look puffy. Nope, they've got edema. I've given this talk to the community, and I've always had some elderly women tell me, well, that's not such a bad thing, is it? Increased intracranial pressure. Major issue are ocular changes and vision problems that are unique to space travel. Due to this pressure, visual acuity is an issue. So a NASA-sponsored survey of 300 astronauts between 1989 and 2011 showed about 29% of short flight meaning less than 3 months, and 60% of long flight, back then a long flight was 6 months, experienced degradation of visual acuity. So most of the time with these short flights, such as the Apollo and the Space Shuttle missions, which lasted about 2 weeks, the changes went away once astronauts were back on Earth. And in these short flights, NASA has figured out how to take care of it. They've got these space anticipation glasses. They can serve as their own optometrist, resolve their issue of visual acuity and be on their way, and when they get back, everything goes back to normal. But there's bigger problems. Problems are swelling, optic disc edema, choroid folds. The choroid is the vascular area of the retina, of the eye. There's swelling in the optic nerve. And these images here on the right, this is a normal choroid. After astronauts have been on flight, they get these folds in their choroid. This can cause lots of issues with vision as well, visual blurring. So I'm going to give you some information of what's up in the International Space Station in case you're interested in doing some studies so you know what can be measured. One can do OCT, tomography, to image the retina and measure choroid folds and other issues with the eye. NASA's launched a huge ocular health study in which this is being studied on now the astronauts who are going up on missions 6 months and 1 year to understand better what's happening up in space. A retrospective study of what was then the longest missions, 6-month missions of astronauts. Of 7 astronauts found, 5 out of the 7 had disc edema, 5 had globe flattening, 5 had choroid folds, 3 had cotton wool spots, 6 had decreased near vision. This is a continuing problem. We'll get back to that with some data from the twin study. Another is cardiovascular issues. And this is a good review that came out fairly recently in cardiovascular research. So lots of issues cardiovascularly as well. Orthostatic intolerance, cardiovascular dysfunction. We'll talk about all of those. Many reasons. There's the direct hits, the fluid shifts, radiations that we mentioned, baseline risk factors, astronaut age, gender, ethnicity maybe, all sorts, any type of inherited factors, indirect hits, occupational hazards such as poor sleep, stress, circadian rhythm disruption. Some of the documented cardiovascular changes in spaceflight related to duration, here they're comparing short duration to long duration. And short duration are those couple of weeks flights from the space shuttle missions. So we've got 13 astronauts short duration, 4 long duration, and they looked at basic measures, systolic blood pressure, diastolic blood pressure. Both of them went up as, kind of a dose response with the time and space. Stroke volume cardiac output decreased, also kind of with the time and space. What we were interested in the twin study was an observation of the thickening of the carotid intima media. Now, astronauts are healthy, they exercise, they're young, some of them. They go up in space and they come up with things that look like atherosclerosis on Earth. So that's not good. Up in the space shuttle, we're able to measure CIMT. There's an ultrasonographic machine up there. And if anybody, so this is the same machine that's used on Earth. The only difference is that here, the machine, you got the probe, and the probe is attached to a computer. Same as the Earth instrument. The only difference is that the buttons are color-coded. So what happens is we sit in, group sits in mission control here. Here's all of us, including Chris and Susan, up near mission control. So the cardiovascular group sits in mission control, and they're monitoring while the astronaut is using the probe to measure their CIMT. And they'll tell the astronaut, okay, hit pink 1, green 3, move your probe to the left, move your probe to the right. And then they get the images. Well, when I was down in mission control in the cardiovascular lab, I noticed all these squidgy balls, and it was kind of really odd to see in mission control, all this technology and a whole desk full of squidgy balls. And I was like, what's with that? And they were like, well, there's a 7-second delay between Earth and this International Space Station. And by the time the astronaut gets the perfect picture and they're able to say, yes, snap the picture for us, the astronaut moves the probe. And so every time that happens, they get frustrated and they throw a ball against the wall. And this system works great. This is my image here, the one on the right. So I wasn't on the space station, obviously. I was just in the next room, so I got a really nice picture of my artery. Okay, another issue is this problem with orthostatic intolerance. So we saw the video that Chris showed that Scott Kelly, our study subject, landed on Earth and gets carried away. There's a reason for this. Not just because of muscle weakness, but what happens is we have arterial blood pressure, we have a gradient when we're on Earth. When we go to microgravity, that gradient goes away. We don't need to pump blood up to the head anymore. Our little muscles around our veins are doing the pumping, gets weak. The astronaut gets back on Earth, their muscles aren't quite ready to keep the blood pumping up to the brain. And so often astronauts might experience syncope. There's many different reasons for orthostatic intolerance that all contribute to the etiology of orthostatic intolerance. There's the decreased plasma volume that we talked about, decreased exercise. Astronauts work really hard. They also exercise as much as they can. But without gravity, no matter how much they exercise, it doesn't equal to what it would be up in space. There's vestibular disturbances, circadian rhythm disturbances, and other stressors, all contributing to this orthostatic intolerance. Gender also plays a role. All of these issues are collectively known as spaceflight-associated neuroocular syndrome, or SANS. When we started the twin study, it was called VIP, V-I-I-P. So if you look at the literature, you'll see VIP before 2016, and then it will be SANS from now on. And then good review is by Stuart Lee, one of the 10 investigators for the NASA twin studies. There's also space sickness. You don't hear about this too often because it's not cool to throw up, I guess, right? And so, but just like us, when we get on a ship or a plane and get boozy, so do the astronauts. There's obviously musculoskeletal issues, muscle atrophy, bone deconditioning. There's no gravity up there, so as much as you exercise, you're not putting the impact on your skeletal muscles. Okay, so musculoskeletal issues, cardiovascular issues, ocular issues. Didn't talk too much about neurocognitive issues, but those are there too. Does that sound familiar? Spaceflight is a model for accelerated aging. Now you've heard that 3 times. So all of you who have heard that going up in space makes you younger, not a good idea. Okay? So all these issues, we need countermeasures for. NASA needs countermeasures for. So NASA has 5 elements in their human research program. One of them is called the Human Health Countermeasures element, and that's responsible for understanding the normal physiological effects of space flights that I just talked about, and developing countermeasures to those with the detrimental effects on human health and performance. And all of these studies they're not just done by NASA people, they're done by scientists like you and us. There's calls, and I'll show you where to go to find research opportunities. Okay, so one of the countermeasures is something called the Chibis suit. So the Chibis suit, and that's me holding one up, they didn't let me get into it because they're so fragile and they didn't want me to break it. It's called a lower body negative pressure suit. So it's essentially a vacuum at your feet that's pulling the fluids back to the feet. And astronauts, before they come down for Earth, a couple of weeks before, they'll do exercise training in the Chibis suit to get ready and decrease the chances for orthostatic intolerance, and also help build up their muscle strength. And something just fun that I wanted to point out is I'm here at NASA in the cardiovascular lab, and just notice just the tiles and the wall and stuff. Whenever we see movies, we see NASA and everything's all like modern and crazy. No, this is a federal government institution. This is like being in the VA hospital or Veterans Administration, no difference. Okay, so the Chiba suit, you know, that was the Russian version. At UCSD, one of my mentors, Alan Hargens, and his student, Niki Ashari, they actually developed a more mobile lower body negative pressure gravity suit for long duration space flight, and that was done at UCSD. So these suits are getting better. Okay, and this is our astronaut, our study subject, in wearing this suit and doing some of the experiments for our study. Okay. So another issue is drug response in space, and another physiological issue. Okay, so here is a review article from 2019. There haven't been a lot of studies published on pharmacodynamics, pharmacokinetics of drugs, but the last time, a couple years ago, I was at the International Space Human Research Conference in Houston, and I started seeing posters on this, so you'll start seeing publications soon. But this is a review of flown studies on human and rodents, and ground analog studies that they do on Earth, bed rest, which I'll talk about a little bit later, and hind limb suspension on rodents. So they keep the mouse, you know, tilted with their hind feet up, and so that simulates the fluid shifts down to the head. So what they've found is in the medications highlighted in red, a significant alteration in pharmacokinetics and pharmacodynamics during spaceflight, both in spaceflight and the ground analog study. Medications highlighted in blue were anecdotally reported to be less effective during spaceflight compared to before or after spaceflight. And that's based on the percent of doses required to be effective. So whereas the studies in red were more controlled, and that those include scopolamine, acetaminophen, that showed PK and PD alteration, the ones in blue come with a caveat. These are self-reported by the astronaut to their medical officer. Now, sometimes when an astronaut goes up in space, they experience new things like fluid shifts. So they may feel that they're congested, and they may take medication for congestion. And it doesn't work because they're really not congested. They're having an experience that's related to fluid shifts. So that's some of the issues with, you know, the self-reported stuff, papers. The other thing is more evidence comes from the mouse studies, the flown rodent studies, where they've seen observed enzymes that are responsible for drug metabolism that are altered in activity during flight or in the ground analog studies. Okay, so you've heard a lot about the physiological effects of spaceflight. But astronauts, whereas all astronauts are going to experience fluid shifts and this hypovolemia, not all astronauts respond the same to the environment, and not all astronauts respond similarly to countermeasures for these maladies. There's gender differences in spaceflight that my colleagues mentioned. The problem with gender differences is there's not enough data yet, because as you may know, that in the 1960s, women were barred from the Apollo program, whereas the Soviet Union was fine putting women to space. It wasn't until 1978 until there was the first female NASA astronaut candidates. In 2007, even when Peggy Winston became the first woman commander of the IS, the International Space Station, in that era, only 10 to 20% of the astronaut corps were women. And it was in 2013 where the astronaut class was 50-50, male and female. So data will be coming out soon. So there's a huge knowledge gap. What factors contribute to human variation in response to spaceflight? Well, omics, you've seen this picture before a couple of times now. Omics allows us to also address not just the physiology, but it allows us to see how molecules in the human body respond to spaceflight. And then when we can integrate the omics data with the physiological studies that I just showed you, we can identify biomarkers to predict the development and progression of spaceflight-associated maladies, and also to monitor the effectiveness of countermeasures. And so it was the NASA twin study that really was the landmark study that piloted human omics studies in the International Space Station. And it can help address really important questions. So this is an example of a hypothesis that was put forth by Scott Smith and Sarah Zwart. So they were one of the 10 groups of the NASA twin study. And Scott Smith is he's the director at NASA, NASA employee director of the nutrition biochemistry group there. So he does a lot of research outside of just our twin study. So they proposed a multiple hit hypothesis of how ocular changes occur in spaceflight, akin to the multiple hit hypothesis for cancer. So there's the stressors of being up in the International Space Station: carbon dioxide, radiation, fluid shifts, some of the arterial stiffness, and then something I didn't mention, but they're working on is carbohydrate and insulin resistance, which is seen in astronauts. All of these contribute to endothelial dysfunction, fluid leakage. Then testosterone, in addition, is another hit. And then finally, what Scott and Sarah are observing is some evidence for inherited differences contributing to endothelial dysfunction. And this comes through the one carbon pathway. Decreased vitamin B status can decrease nitric oxide, which, as you know, is important for vascular tone, creating increased oxidative stress and increasing endothelial dysfunction, more fluid leakage, edema, goes down the line, and impacts the eye. So omics studies, genetic studies, gene expression studies, epigenetic studies, all can contribute to understanding this pathway. Okay, so the one-year mission in NASA twin study. So I'm going to give you some data that's integrating molecular data that we obtained with the physiological data, coming not only from the twin study, but the parent study, which was the one-year mission, which was the study with Scott Kelly and his Russian counterpart, cosmonaut Koryenko. So we have, some of my co-investigators in the twin studies were also participating in the one-year study as well. So we have data that we can integrate from both studies. And by the way, working with astronauts is great because not only are they great study participants, they do all the phenotyping and sample collections for you. Okay, and so they obtained all the samples from every tissue you can imagine for us, which was great: blood, urine, stool, saliva. And we were able to get a lot of data. Okay, so ocular changes with space flight. So we did some of this OCT studies on both the ground astronaut and the space astronaut. I'm trying to be IRB good and not put names out, but you know who they are. Okay, so these studies were led by the Cardiovascular and Vision Laboratory. My co-PI in the NASA twin study was Mike Stenger at NASA, who's the director of the cardiovascular lab. Brandon Macias was our postdoc at UCSD, who then later got a job at NASA and led the ocular studies for the one-year mission, and Stuart Lee, who is another one of the principal investigators for the NASA twin study. So what we showed here is we were interested in choroid thickness. Remember those choroid folds in the eye? And retinal thickness. So what we saw is, again, blue is the space twin, green is ground control twin. What we saw for both retinal thickness and choroid thickness was a jump during space flight. And then after landing, these thickness kind of returned to baseline. Whereas the astronaut who stayed on Earth, there was no difference, there was no change really throughout the pre-flight, flight, and post-flight time points. Following up the twin study. So I always talk about this and then say, well, post-flight, you know, there was still a little elevated thickness, but what happened, people ask me what happens a year later, does it continue? Well, that data came from the one-year study, not my study, but the study that was led by our then postdoc, Brandon Macias, who became a senior investigator at NASA. So what this data showed, they were looking at both Koryenko and Scott. What they saw was these choroid folds in both astronauts progressively worsened throughout the one-year mission. The longer they stayed up there, and the worse it got. The maximum height, so they look at the height of the folds, and that's the measurement. The maximum height did decrease post-flight, but it didn't return to pre-flight values by 180 days or the 365 days flight examination. So there were still choroid folds there, just much less than there was in flight. They also looked at edema, and this is edema in the right eye, at flight day 30, and it persisted until 90 days after return to Earth. So this is just with the one-year mission that they're seeing persistence. Imagine what could happen after 3 years in space. We were also looking at fluid shifts-related changes. Looked at internal jugular vein area and forehead thickness. These measurements were taken seated and supine in both our space astronaut and ground control. And what we can see here is that when the astronaut went to space, their jugular vein area and forehead tissue thickness was increased either to or above the level of supine on Earth, but it returned somewhat to baseline levels after they returned to Earth. Also, a rapid increase in carotid intima media thickness after launch, with the compensatory increase in carotid artery diameter. And you can see in CIMT, after the return, this was only 4 days, that was the last time point we had, it was still increased. So the question is, are these changes reversible once they happen, or do they represent a lifetime of risk, cardiovascular and cerebrovascular risk for the astronauts? Okay, so we were also interested in relating it to the molecules we were studying. One thing we looked at were inflammation markers. So this is Scott Smith's inflammation markers. And so we found that some of the interleukins and prostaglandins, when there was a jump in carotid diameter and CIMT, we also saw increases in these indices of inflammation as well. More studies are being done on more astronauts to make sense of all of this data that, was the first kind of pilot data that we had. Okay, so we had sampling, both plasma and urine samples from all these time points. So what this shows is pre-flight, post-flight, and return sampling, 24-hour urine collections on the first row is spaceflight astronaut, and the next row is our ground twin astronaut. So we have a lot of time points, and I'm just going to show you some of our proteomics data in relation to some of our physiological measures. So we did untargeted and targeted proteomics on the urine, and we saw collagen alpha 1 shedding in the urine during space flight. We also saw apolipoprotein shedding during space flight. And in the second column here, you can see late in flight, there was a lot of, let me go back. This is urine, and this is plasma data. So in the plasma, we saw an increase in apolipoprotein. In the urine, we saw a lot of shedding of collagen. So, there's a lot of hypotheses about this. For the shedding of the collagen, we think it might have something to do with vascular remodeling. Okay. The other thing that we noticed was an increase in lactic acid and pyruvic acid ratio during flight, which indicates a shift from aerobic to anaerobic metabolism. And there could be many reasons for this. One of the reasons we thought was perhaps, it could have been the weight loss during flight, muscle catabolism, or loss of mitochondrial function. So with Hemal's lab, we looked at mitochondrial function. So we used the seahorse assay in his lab to see if blood samples from the different time points differentially drive mitochondrial function. And so what we did was we had different, and when I say we, I mean the postdoc, Jamila, who did all this tedious work. We had cell lines, muscle, heart, immune cells, and we spiked it with plasma from the astronaut and the ground control at different time points. And then we measured mitochondrial oxygen consumption rate under varying conditions of uncoupling mitochondrial pathways by putting drugs in, and that's the whole seahorse assay idea. So what we found was in the muscle cells, during flight, increase in basal respiration, increase in ATP-linked respiration. In flight, spare reserve capacity decrease, non-mitochondrial respiration decreased. So there was definitely some evidence for mitochondrial changes in flight. And with Chris Mason's data, Chris Mason's RNA-seq data showed higher levels of mitochondrial RNA in flight. So this is all kind of interesting of what's going on with mitochondrial function in space. Okay, and so I'm going to end with ground analog studies. Okay, so there's only so many astronauts that go up in space. So one can create a simulated space environment and study them, humans, on Earth. And so this is a big area at NASA, and they're called analog missions. So to experimentally simulate this fluid distribution, you put a person supine and tilt them 6 by 6 degrees and you leave them tilted for 30 days, 70 days, and they have to do everything in this tilted position. So there's been a number of what's called 6-degree head-down tilt bed rest studies, and this is sort of the details of how they do everything. In the past, they would have to eat tilted, but they were able to use their elbow and kind of, wedge themselves up. And this is a twin study, bed rest twin study where they did, which was discordant for exercise. That was done at UC San Diego. So these were all, different studies that have been done in the past for simulating the microgravity. And while they're doing the head-down tilt, they're also analyzing the effect of countermeasures on some of the issues that the astronauts will face in microgravity. So one of the things they were looking at was changes in stroke volume, which is represented here on the top of that diagram there, and changes in internal jugular vein diameter. So you can see that when a person's upright, my thing is not working here, but when a person's upright, they have a small jugular vein diameter. Supine gets a little bigger and this was a 15-degree head-down trip, gets even bigger. Now they were interested in the countermeasure of what happens if you put someone in a chibis suit when they're head-down tilted. Well, this chibis suit actually kind of helps with that jugular vein increase, and it kind of decreases it and also decreases the stroke volume as well. So that's some of the ways that they're using ground studies to see what countermeasures may work in space. I was involved in this study and called the Vapor Study, which was actually conducted at NVHAB, which is operated by the German Space Agency. And I'm checking how much time we have. Getting time, right? So I'm just going to go really quick. So the difference with this is we had 11 subjects, 6 men, 5 women. Very stringent, very stringent. No more hanging out on the elbow to eat. They actually mimicked the carbon dioxide level. They weren't able to use a pillow. Okay, so this was the best data that had come about to that time. It actually simulated some of the effects and the ocular effects, which had never been seen before with these studies. So we looked at metabolomics, pre, during, and post. And what we saw are different kinds of patterns. One of the patterns we saw in the comparison of metabolite levels versus baseline versus, during the different time points was some groups of metabolites changed immediately during that postural shift to the head down position and remained increased, but then gradually came back to normal baseline levels. And then when the subjects were put back in normal posture, 30 days later, those metabolites shifted up again. So that was interesting. Some of them, you can see, some of them had some of the metabolites had long-lasting effects, some of them had less. As you go down, they have longer lasting effects. As you go up, they had lower lasting effects. Okay, so I'm just going to throw out a couple of, metabolites. One of them was tyrosine, peak during the postural shifts. Well, probably what's happening there is tyrosine hydroxylase increases with postural shifts. And so that is probably metabolizing tyrosine into this acetyltyrosine, which is a precursor for your catecholamines. So that makes sense. Phenolactic acid, also you see spikes on the 2 postural shift days. Well, This is a little bit interesting. What's going on here? We've seen the precursor change with exercise. Well, we haven't, but other studies have with exercise. And so something's going on with this particular metabolite and its precursors, but it's also seen spiked when people eat chocolate. So I'm trying to figure out whether they were given chocolate to cope with the shift on day one and as a reward for finishing, given chocolate again, but I don't have the diet data yet. There were other shifts also. One of them, one of the patterns you saw was an increase upon, gradual increase upon entering head down tilt, and then it just got more and more until the last day and then dropped back to baseline. One of the metabolites that showed this was this methylgluconic acid. When this builds up, that impacts the TCA cycle, so you can get some mitochondrial issues there. I'm going to go fast. The other thing we saw was a spike in the succinic acid. That is part of the pathway that one sees altered during hypoxia, and also part of the mitochondrial pathway. So we've got some interesting data on metabolites that are changing in simulated space-like conditions that are giving us some ideas on how to approach studies up in space. Okay, so correlation patterns we're seeing between metabolites. So that shows that there's pathways that are being disrupted. And pathway enrichment analysis showed a lot of mitochondrial pathways were disrupted in this bed rest study. We've got 70-day head down tilt studies, data and metabolomics, proteomics, and also physiological studies. We also have the twin study data. So putting everything together, all this metabolomics data from all the different cohorts, we can start parsing out what changes might be due to carbon dioxide, might be due to exercise countermeasures, testosterone, so gender differences. And I'm going to end there. If you're interested in NASA studies, this is your go-to place. Go to NSPIRES, and you have to weed through all the information here because there's a lot of calls for grants from physics, biophysics, chemistry. You have to find the human research stuff. Human research roadmap. If you go to the human research roadmap, pull out your favorite disorder or phenotype, and you'll get all the information on what NASA's interested in. You'll have a contact. In this case, it's Stuart Lee, our buddy from the NASA Twin Study. Contact them, and they might be interested in working with you on a project. And if you're a postdoc or a grad student, go to that website. You can do a postdoc with one of the investigators at NASA. All right, I have a lot of thank yous. Everybody that's here, of course, are on this, and NASA Twin Study investigators, Scott and Mark Kelly, and I'll end with that. Okay.
All right, we'll take any questions that you may have.
Daniel Bobek from Czech Republic, very interesting presentation. I would like to ask if there is any medication every astronaut take to countermeasure the spaceflight effects, or what are most common medications astronauts take in space or after returning to home?
As far as the medication, they've got the general medication up there. They got the aspirins, the acetaminophen, the scopolamine, antibiotics. But there's nothing there for the actual space-related stuff. Okay, so when I was, before I did this study, I was doing a study with mice. I was interested in doing gene expression in mice that got flown in space. Well, it happened to be that Amgen was doing a study for bone loss related to space flight. So there are medications that are being developed for space flight. Well, the interesting thing about this bone loss drug is the mice came back on Earth. NASA gave me the mice. We dissected them, did all this work in Florida, brought it home, and then we get a call from NASA saying, we mislabeled the mice. You have Amgen's brains. And so this was a big deal because it was Amgen's drug-treated brain samples. And they made us throw away all those brain samples because Amgen wouldn't allow us to analyze that drug. But I don't know what ever happened to that drug, but it's all in development by pharmaceutical companies.
Thank you very much.
Very good. Any other questions? Well, thanks for sticking it out for the rest of the symposia. Have a rest of a great week.